Good day everyone, welcome to the course on next generation sequencing technologies, data analysis and applications. In this lecture, we will be talking about genome organization in 3-dimensional space. In the last few classes, we have talked about different types of epigenetic modifications and how we can identify those using next generation sequencing technologies. So, we have talked about DNA methylation, transcription factor binding site identification or histone modification identification, and then we also talked about chromatin accessibility. So, those are the modifications on the DNA or some sort of binding of the proteins to the DNA, and they actually influence how they actually influence the gene expression. In this class, we will be talking about the 3-dimensional configuration of the genome itself. Okay, there is no other protein that is actually modifying expression patterns or directing expression patterns. Is the 3-dimensional configuration itself, how it can actually influence the expression of genes. Okay. So, of course, this 3-dimensional configuration, they are associated with some sort of modification which could be nucleosome occupancy, or it could be some other interaction between different elements in the genome, which can then drive the expression. Okay, so, let us start with that. So, this is the agenda for this class. So, we will be talking about organization of genome in 3-dimensional space. Here are the keywords for this class. The first one is TADS and the second one is LADS, and then we will talk about high-seq. So, we will explain these terms. So, as we go along. So, the question that we are interested in is that how is genomic material organized in 3D space inside the nucleus. Okay, so, the genome, right, is not like compressed in one location and everything is all mixed up together. Okay. So, it also has a 3-dimensional configuration, okay, and this is something what this study is all about. Okay. And this 3-dimensional configuration is very important for long range interactions, right. So, there are different elements of gene regulation. So, you can have, for example, enhancer that interact with promoter and can influence the downstream expression expression of the downstream gene, right. So, this also has regulatory links, right. So, we can have these long range interactions influencing gene regulation, and one example that I gave is the enhancer promoter interactions. And if we understand this genomic organization in 3D, we can then map these enhancer promoter interactions very accurately. Okay, so, the earlier method, so there has been a lot of interest in studying this 3-dimensional configuration of genome or this interaction between different genomic regions, and the traditional method utilized something called FISH or fluorescence in situ hybridization. Okay, so, very briefly, this method utilizes a fluorescent probe for identifying this location of sequence in the genome. So, let us say you are interested in a specific sequence. So, you will design a probe that is complementary to that sequence, and of course, you also have the fluorescent molecule that is attached to this probe. So, with microscopy you can then identify the location in the genome where this probe is going, and you can then say, okay, this is where the probe is there. So, you can utilize this method to actually study interaction between two different regions of the genome. So, let us say your hypothesis is that this region A is interacting with region B, right, and then what you do is then you want to test this, right. So, whether region A, region B, they are interacting with each other. So, you design probes against these regions, right, region A and region B with different fluorophores, right. So, one could be green fluorophore and there could be red fluorophore, and then you can see their localization in the genome, right. If you see that these probes are these fluorescent molecules under microscope, they are localized to the same region of the genome, it is likely that they are interacting. Now, as you can realize that this is probably quite time consuming method, right. So, you have to design probes for each sequence, and then of course, you cannot actually scale it up, right, for studying all sorts of interactions that are happening in the genome, right. So, you cannot apply it in large scale across genome wide. Okay. So, as I mentioned, this is low throughput, but if you are interested in looking at interaction between two specific genomic regions, right, and these are of interest to you, then you can of course apply this method and you will get some sort of idea about the distance in 3-dimensional space in the genome. Okay, so, before we actually go into the methods that allow us to do this in high throughput manner, right, what I will do is I will introduce certain terms that are commonly used, and then I will discuss the methods. Okay. This way it will be better for you to understand, because without understanding these terms, if we use those later on, that will be very confusing for us, right. So, let us introduce these terms first and then we will discuss the methods, but actually in reality this happened the other way, right. So, researchers applied those methods first and they identified these different regions of the genome and they use these terms, right. But we will go in another way for a better understanding. Okay. So, the first term that will come across is called chromosome territories. Okay. I will explain this in a moment what chromosome territories are, and I will also have a schematic diagram for a better illustration. And then the next term we will use is called A B compartment. So, A compartment or B compartment, we will use the term called topologically associating domains or TADs. This is a term that you have seen in the keywords. Okay. I will explain what these TADs are. Then you have lamina associated domains or LADs, again you have seen this in the keyword. Similarly, you have something called nucleolus associating domains or NADs, and now we will discuss each of these terms carefully. Okay. So, the first term is chromosome territories. Okay. So, it turns out that in a genome, right, let us say human genome, the chromosomes actually occupy specific region in the nucleus. Okay. It is not like all chromosomes are jumbled up or mixed together. So, each chromosome occupies a specific region in the nucleus and these are called chromosome territories, right. So, you have chromosome 1 located in specific location, chromosome 2 may be another location, right, and chromosome 3 and so on, and which means there is limited spatial interaction between two chromosomes. Okay. There is some interaction, of course, you can look at some of the data that I will also mention in the reference, and you will see there is some interaction between certain chromosomes, but these interactions are quite limited. The next term is AB compartments. Okay. So, what researchers have seen is that chromosomes are organized in two compartments, one is called A compartment, the other is called B compartment. So, A compartment is associated with open chromatin and this is transcriptionally active region, right. So, we have now explained or come across this term open chromatin many times, and you understand, right, this region is open for transcription factor binding and RNA polymerase. So, transcription can happen, right. So, this region is transcriptionally active. On the other hand, you have B compartment which is associated with closed chromatin. So, these are all so this region is condensed, right. So, it is not nucleosome occupied and mostly transcriptionally inactive. The next term is topologically associating domains or TADs. Okay. So, these are regions in the genome that preferentially interact with each other. Okay. So, if you look at the 3-dimensional structure, you will see these regions, they have lot of interactions within themselves, okay, and they have much less interaction with other regions. Okay. So, these regions are called TADs and they might have regulatory role and which can impact the gene expression pattern. One interesting point is that TAD boundaries, these are marked by CTCF, okay, binding site. So, this is a transcription factor that recognizes a specific sequence, and this has been seen that these boundaries or the ends of TADs are marked by this CTCF binding site. We will see the figures in a moment, images, some sort of illustrations that will be better for you. And also there is presence of cohesin, it forms like a ring like structure and that kind of keeps the kind of like these TADs kind of intact. Then you have lamina associated domains or LADs. So, these are regions of the genome that actually interact with the nuclear lamina or specifically to lamin protein. Okay. So, what it means is that then it is mostly kind of associated with the nuclear lamina, right, and this is mostly transcriptionally inactive. Okay. Now, there are two parts of these LADs. So, you have some constitutive LADs or CLADs. So, these are in contact with lamina across cell types. So, if you study these regions across different cell types, you will see like in all cell types these regions are in contact with lamina, and these are mostly closed chromatin and they are transcriptionally inactive. And then you have something called facultative LADs or FLADs. So, these are regions that are in contact with lamina in some cell types and they are not in contact in some other cell types, right. So, this is kind of not like constitutive LADs. Okay. Then you have the final term, which is the nucleolus associating domains or NADs. Like LADs, these are regions that interact with the nucleolus. So, nucleolus is the region of the nucleus where ribosomes are formed, right. So, this is where the nucleolus interacting regions, right. So, these are also mostly heterochromatin in condensed form, and they are transcriptionally inactive. Okay. So, what you have seen, CLADs and NADs are mostly transcriptionally inactive. FLADs can be transcriptionally active or maybe inactive. So, just to illustrate better, I will use a schematic diagram, some animations, right, so that you understand this better. So, imagine this is the nucleus, right, the circle is the nucleus, okay, and inside the circle you have those different colors, right, ribbons of different colors, these are the chromosomes. Okay. So, each chromosome is occupying its own space. So, these are chromosome territories, right, and you can see that there is limited interaction between the chromosomes. Okay. So, you can see some interactions perhaps here, right, or maybe here, but in total, like this is quite limited. Now, if you zoom in, right, in one of the chromosomes, you will see like A and B compartments, and just to remind you again, so A compartments are transcriptionally active, which means mostly open, and then you have the B compartments that are in condensed form and they are transcriptionally inactive. Now, if you zoom in further, right, in the small region here, what you will see are the TADs now. Okay. As I mentioned, TADs, these are topologically associating domains, right. So, they interact preferentially within themselves. So, here is a TAD, here is another TAD, right, and you see this, they are interacting within themselves more, okay, and they are not interacting much with other TADs. Okay. So, this is actually the definition of TADs. Okay. And if you zoom in further, you will see these chromatin loops and these TAD boundaries, right, where you have these CTCF proteins. So, you can see this protein egg-shaped proteins, right, this is CTCF, and then you have ring like structure which is formed by cohesin. Okay. So, this actually kind of like keeps the TAD in place, or maintains the structure of the TAD. Okay. Of course, there are a lot of studies going on, but this is kind of the working model right now. Okay. So, how do you study this genome organization in 3D space, or how did we come to know about all these terms, all these aspects of genome organization, right. So, TADs, LADs, etc. Okay. So, what kind of method did you use, or did researchers use to actually understand that? And this is something what we will discuss now. Here are several methods that I will discuss very briefly. So, we will start with the 3C and then the other methods that are based on 3C, and then there is the final method, which is the CHIA PET method. Again, we will discuss very briefly all these methods. So, 3C is the first method that was actually devised to study this 3-dimensional genome configuration. Okay. So, the full form of 3C is the capturing chromosome conformation, right, as you can see. So, it is called 3C, and this is the reference paper that appeared in 2002. Very briefly, we will discuss the steps, how this works, and then of course, we will look at the different variants or the different 3C derived methods, those 4C, 5C and high C, right, and we will talk about their advantages and disadvantages, and finally, we will talk about the CHIA PET or CHIA PET method. Okay. So, the first step is cross-linking by formaldehyde. Okay. So, we have come across this before, right. So, imagine these two regions, you have the blue and the red region, right, they are interacting here, this is shown in this circle, right, this is the region where the interaction is happening, and before we prepare the sample for sequencing, we want to preserve this interaction, right. So, to do that, we do the cross-linking by formaldehyde, and this cross-linking actually preserves that interaction. Okay. And this cross-linking happens mostly due to proteins, right. So, this is protein mediated cross-linking, the proteins that are present in this region, they will form this cross-link. Once you have done the cross-linking, so the next step is restriction digestion. So, you have seen you see like I have highlighted some of these restriction sites, okay, and this is where you can do restriction digestion. So, these sites can be same or different, right, again depending on your experimental design. So, the next step is actually intramolecular ligation. Okay. So, once you have isolated this fragment, right, by restriction digestion, the next step is intramolecular ligation. So, what we will do is we join one end here. Okay. Now, once this end is joined, then we actually reverse the crosslink, we remove this crosslink, and then the DNA is linearized. Okay. Now, the next step is once we linearize the DNA, we can then amplify, right. We can use these primers in black, and we can detect by PCR amplification or by quantitative PCR, right. So, if two regions are interacting, you will see a PCR band or you will see a signal in quantitative PCR, and then you will infer, okay, there is interaction between these two genomic regions. Now, as you realize, right, this method can be applied when you have a hypothesis, right. So, you want to investigate interaction between two genomic segments. You know, okay, let us say you hypothesize let region X and region Y, they are interacting, okay, and then to test that you apply this 3C method, you design primers that bind to this region X and region Y, and then you do the 3C, and you see whether there is some sort of signal in the quantitative PCR at the end, okay, and based on that you draw inference, right, whether there is interaction or not. So, what you also realize, this is mostly mapping one-to-one interaction, right. So, we are testing interaction of one genomic region with another genomic region. Okay. So, this is one-to-one interaction, and the scalability is poor. So, we cannot scale this up very easily across a large number of genomic regions, right. So, if you want to test this for 1000 regions, right. So, maybe just one region with 1000 other regions, this becomes very difficult to do, right. So, you have to design these primers, you have to do all the quantitative PCRs, and that becomes really tedious, okay. And 1000 is already a small number, right. So, for a big genome like human genome, you can have millions of these interactions, and you cannot scale this up, okay. And then also the results are dependent on restriction enzymes that we use to some extent, but then you can counter this by using different restriction enzymes and looking at the interaction patterns. Okay, so, to counter this scalability issue and this one-to-one interaction, so the question is then, can we identify genomic regions that interact with a specific genomic region? So, specifically the example that I mentioned, right. So, we take one genomic region and we want to test whether there are 1000 other genomic regions that interact with this region. Okay. So, this is something would be very interesting, like let us say we have a gene that is very important for some purpose, and then we have the promoter of the gene, and we want to see whether the promoter region of that gene interacts with other genomic regions which might influence the expression of this gene. Okay. So, maybe instead of testing these 1000 other genomic regions, what we can do is we can ask whether we can explore, right, or we can unbiasedly discover regions that are interacting with that genomic region. Okay. So, this target region or this promoter that I just mentioned, this would be the bait or the viewpoint region. Okay. These are the terms that are used in this analysis. Okay. And this is a method that actually can enable us to do that. It is called chromosome conformation capture on chip, or 4C, and this is the paper that actually described this method in 2006, and this method allows for an unbiased search for genomic regions that interact with a specific locus. So, this specific locus is termed as bait or viewpoint. So, here are the steps for this 4C method. So, the first few steps are exactly identical with 3C, right. So, we have first step is cross linking. So, we want to preserve this interaction. The second step is restriction digestion, right. We are taking this fragment out from the genome, right. So, we are generating these fragments, and then we have ligation, right. So, we are generating this or ligating these fragments back, but then the difference starts. Okay. So, after this ligation we cross link reversal. So, we remove the cross link, we linearize, okay, and then we have a secondary digestion with another restriction enzyme. Okay. So, I have marked these restriction sites by these black bars. Okay. The difference from the first restriction digestion is that this second enzyme is a more frequent cutter, means it will cut more frequently in the genome. So, usually the first restriction enzyme is a 6 base pair cutter, right, which are rarer in the genome, and the second one is 4 base pair cutter. So, these are much more common in the genome. Okay. So, we take a second restriction enzyme to actually do the digestion. Okay. So, the idea is to generate smaller fragments after this restriction digestion. Now, once we have these smaller fragments, we can then ligate back, we can circularize these fragments, right. So, we can ligate these ends and we can circularize this fragment. Now, what one thing you probably realize, right. So, we are doing this in parallel for different genomic regions, right. So, instead of just starting with this one interaction, we have multiple interactions, and for each of them we will have this kind of circularization, this digestion, etc. So, at the end of circularization, we will get these different types of fragments, right. I am showing them in different colors. So, these are the regions that interact with the red region. Okay. So, here the red region is the bait or the viewpoint. Now, once we have generated, we know the bait sequence, right. So, this is the specific locus that we are interested in. Okay. So, we can then design primers on the bait, right. So, here are the bait sequence, right. I can see these primers here in black arrow, and we can do something called inverse PCR. This is not normal PCR. You can see the primers are pointing away from each other, right, and this is inverse PCR, which will actually generate these amplified fragments containing the other fragments, right. So, other these blue regions or green regions. Okay. So, these are the interacting regions with the bait. Okay. So, once we have generated this, now we can then simply detect by microarray. So, earlier we used to detect by microarray, now we can also say we can apply a next generation sequencing here. Okay. So, there is a variant of this 4C which we will discuss again. So, just to summarize what we have discussed. So, we have to again compare with a control because this is a microarray experiment. So, we will get some sort of enrichment which we will have to compare with a control. Okay. So, in this case the control is restriction enzyme digested genomic DNA, right, and then we see enrichment of specific regions compared to the control that indicate interaction with the bait region. There is a variant that, as I mentioned, is called 4C or 4C sec. The full form is slightly different, it is called circular chromosome conformation capture, or sometimes also referred as circular 3C. Okay. And this is the paper that actually proposed this method. So, the protocol is slightly different. So, let us look at that. So, the first step is cross linking as before, the second step is restriction digestion, okay. And the third step is circularization. Okay. So, there is no linearization, then a second restriction digestion. So, this is actually a much simpler process. Here we have circularization. So, we circularize this fragment. Now, you can again imagine that this is happening for multiple interactions for the bait, right, and we get these different circular fragments, and we can again do the inverse PCR directly, right. And once we have done the inverse PCR, we can then detect by macroarray, or we can apply NGS methods to actually identify these regions, blue green regions. Okay. So, this will tell us which are the regions that are interacting with the red locus or red region. So, what you see from this discussion is that this is 4C is one to all mapping, right. So, we have one bait or viewpoint sequence, and we are looking at interaction of this region against all genomic regions, right. So, we are identifying genomic regions that interact with this bait region. Okay. So, this is an one to all mapping. Now, the idea is like, can we extend it to many, right. So, instead of just having one bait, can we apply this kind of method to many baits, right. So, can we do this in parallel for multiple locus, not just one locus. Okay. And this actually led to this method called chromosome conformation capture, carbon copy, or 5C method. Okay. So, as you can see, we are getting 3C, 4C, 5C, and then finally we will get to high C. Okay. So, this is a many to many mapping, okay, and this is the paper that actually suggested or proposed this method. So, let us go into the process how it is done. The first few steps are exactly identical to 3C, right. So, you can see the cross linking, the restriction digestion, and ligation. Okay. So, you can start with the 3C process, and then you can try 3C or 5C, right, depending on your need. So, here the difference is that instead of the linearization, etc., so linearization happens, right. So, after the linearization, what happens is something called multiplex oligoannealing and ligation. Okay. So, what is this multiplex oligo? So, you have this pair of oligos, right, that can bind or recognize different regions, okay, of the genome, and then they can anneal to those fragments, right, of the genomic regions, and also can ligate with each other, right. So, there is this ligation between the oligos. Okay. So, what will happen, because these are multiplex oligos, mean they contain a combination of oligos, right, different oligos that can bind to different genomic regions, and so they will anneal, right, based on their complementarity to different genomic segments, and they will also anneal and then they will also ligate. They are designed in such a way that they will ligate with each other. Okay. So, once they ligate, right, so then what you can do is you can then amplify with universal primers. So, what you see is that even though they contain these multiple oligos, these N regions are identical for each of them. Okay. So, these are the regions where we can use for universal primers, primers that will bind to all of these combinations, okay, and then we get these amplifications, which we can then sequence by next generation sequencing, or we can identify by microarray. So, what you have seen, this is many to many mapping, right. Why aren't we saying this is all to all? Because we are limited by the multiplex oligos. We can contain only a certain number of combinations in the oligo library. Okay. So, we cannot have all oligos in the library. So, this process is limited by this oligo design. The final method that we will talk about is Hi-C in detail, right. So, this is an extension of the earlier methods. This is all to all mapping, and this is the paper that actually proposed this method. So, here is the protocol. So, we start with cross linking, we then do the restriction digestion. Okay. The next step is something called end filling, that happens at the end of these fragments, okay, and a biotin marking. Okay. So, this is a chemical molecule that is attached to the DNA, okay, because this will help in further purification of the DNA fragment. Okay. So, the next step is intramolecular ligation. We ligate these fragments using ligase enzyme, we fragment, and then we pull down this biotinated DNA using a molecule called streptavidin. Okay. So, biotin streptavidin interaction is very strong. So, this will pull down this biotinated DNA, and once we have these fragments, we can then separate out these fragments, and then we detect these fragments by NGS. As you can see, this is all to all mapping, Hi-C, right, because we are not limited by any oligo design, etc. We will get all sorts of fragments that are marked with biotin, and they will be sequenced by the next generation sequencing method. But it is dependent on restriction enzyme that is used, but then this is easy to overcome, because you can do the experiments with different restriction enzymes, and then you can compare the results and see if you get similar results. And this has been applied for mapping interaction within chromosomes and between chromosomes in the human genome, and here is the reference you can look up. Now, one of the things that we will get from the Hi-C map is strength or frequency of interaction between genomic regions, and this will look something like this. If you have the genome, for different genomic regions, you can see the interactions by different colors. These interaction strengths will be represented by different colors. So, darker color means stronger interaction, lighter color means weaker interaction. So, as you can see, there is this region that shows very strong interaction within itself. So, this is likely to be a TAD. Here also you have this other region that shows very strong interaction within itself, and this probably is a TAD, right. So, just to interpret this square here, this actually looks at interaction between this genomic region and this genomic region, right. So, you can then interpret this across all these squares, and you can look at interaction between different genomic regions. So, you can overlay this along with ChIP-seq data. So, you can do ChIP-seq on the same sample, and you can overlay with ChIP-seq for, let us say, CTCF binding site, and maybe here are the peaks for CTCF, meaning we are looking at TADs, right. So, remember the TAD boundaries are marked by CTCF, right. So, this actually helps the information of the TADs. You can also look at transcription profile of these regions, right. You can then classify into A compartments, B compartments. You can also look at histone modification patterns, again by ChIP-seq or ChIP-exo, and you can then actually overlay these with lot of things. So, this kind of helps us in understanding these interactions, okay, and this kind of figures you will see across all the interaction maps that you come across in the references. The final method I will just very briefly mention is called CHIA-PET or CHIA-PET. I will not discuss the protocol. So, the full form is chromatin interaction analysis by paired and tag sequencing. Okay, here is the reference. So, what this method does, it finds interaction sites that are mediated by specific proteins or transcription factors. So, on top of these interaction maps, you also get the proteins or transcription factors that are mediating those interactions. So, this is, you can think of this as Hi-C coupled with ChIP-seq. So, this Hi-C method, this is so powerful that this has now been extended to single cells, right, and you can get single cell Hi-C. So, and this is the paper that actually shows this cell to cell variability in structure, chromosome structure, right, and this is something that you can actually now study with single cell Hi-C. Another project I will mention is called 4D nucleome project. So, this is looking at 3D organization of the genome and how it changes with time, which is the fourth dimension. Here is the link to the project, and here is the paper for this 4D nucleome project. So, these are the references for this class. To summarize, we have seen the genome has distinct 3D organization, and some genomic regions interact more frequently within themselves than to other regions, and we have seen these are called TADs, and these structure actually impact transcriptional activity. We have discussed different methods, 3C, 4C, 5C and Hi-C. They enable us to detect the interactions between genomic regions in 3-dimensional space. Thank you very much.